The Meet the Researcher online discussion this evening, hosted by the Westmead Institute for Medical Research, or WIMR as it's more colloquially known. I'd like to begin by acknowledging the traditional owners on the land on which we meet and pay my respects to their elders past and present. Thank you for taking the time to log into this COVID-19 themed Zoom evening. It's really important that we stay connected. We can't meet in person, but this is going to be a great alternative, so thank you. We're going to have presentations this evening from Professor Tony Cunningham and Professor Sarah Palmer. My name's Sophie Scott and I'm the medical reporter for ABC, and I'm going to be moderating tonight's session. And we're going to be having two presentations, as I mentioned. They'll give an overview of COVID-19 and the amazing research that's been done. We'll talk about the origins of the disease, the spread of the disease, and importantly, the really good work that WIMR is doing on this. Before I get underway, I'd like to just let you know that your microphones and videos have been muted, so they won't be active during the presentation. So you won't be able to be seen on this Zoom call and ask questions, but at the end, you'll be able to ask questions. You'll be able to write in questions on the chat function. So, and we've already got some of your questions already, which is great. We do have a time constraint this evening, so we won't be able to get to everybody's questions. We'll try to get to as many questions as we can though. We expect that the presentation this evening will go for about 45 minutes. And look, as everyone knows who's listening and watching, COVID-19 has changed how we all live. The fact that we're having to do this session like this and not meet in person, as well as the immense human toll that this virus has taken on all of us. And luckily and thankfully though, the spotlight has really turned on science and scientists to help solve this crisis. And as a leading Australian medical research institute, WIMR is focused on making the world a better place for all of us. And their vision is to lift the burden of serious disease and illness. And the way it's doing that is by cutting edge science that is there to directly help patients. They've got 5 areas of research, but the key one we're going to talk about this evening and we're going to be focused on tonight is infection and immunity. And you can see they do things like HIV, herpes, influenza, MS, type 1 diabetes, kidney diseases, so all number of things. Tonight, you're going to be hearing from Professor Tony Cunningham and Professor Sarah Palmer. They jointly lead the Centre for Virus Research. And one of their key projects that we're going to be talking a lot about tonight is a vaccine for COVID-19, particularly to prevent it in the elderly and older people who we know are the most vulnerable when it comes to COVID-19. The team working on the vaccine is led by Professor Tony Cunningham. He's arguably Australia's leading researcher focusing on vaccine development in older people. He's an infectious disease physician, virologist and scientist known around the world for his work on HIV and herpes viruses. And he made some key discoveries leading to the development of a shingles vaccine, amongst other things. He founded WIMR and was awarded an Order of Australia for his service to medicine and research. So let's now hear from Professor Tony Cunningham. Please give him a virtual round of applause. Welcome, Tony.
Thank you, Sophie. So the origin of the SARS coronavirus type 2, which is the cause of coronavirus infectious diseases in 2019, otherwise known as COVID-19, is clearly bats, and it's most likely that it's been transmitted through mammals to humans, very similar to the first SARS coronavirus, which was leased onto the world in 2002 to 2004. We know this because of the sequencings of hundreds of strains around the world, and we know that bats have 10 coronaviruses, and this is the second to be released into humans. It could happen again. We now know that more than 3 million people have been infected around the world. One person can infect 3 to 5 others, and we need to get down under the 1-to-1 ratio in order to prevent spread. Most spread is symptomatic, some is asymptomatic, and we'll come back to that later on. The average mortality for COVID-19 around the world is 7%, compared with seasonal influenza of 0.1%, but this is variable, higher in Italy, and in Australia, approximately 1.3% at present. We may learn more about this as we do more testing and more antibody tests, and those figures may change. The mortality is higher in aged care facilities, not being properly quantified, but an example is Newmarch House, where the mortality has been 40% at present. Okay. Now, we're very lucky at the precise time at which we're dealing with this terrible pandemic. We've been waiting for one of these for years, and one of our pieces of luck is that we have excellent diagnostic tests that can be ramped up to hundreds of thousands. The way in which the swab is taken here is from the back of the nose. In future, we'll probably be able to take swabs in a more comfortable fashion, and the tests are highly sensitive and can be, as I said, automated. And there may be new tests coming in the future which are even faster in their turnaround than the current tests. Next slide. And what can we learn by looking at countries around the world? The World Health Organization is looking at this very carefully at present, looking at 170 parameters of what different countries have done. And we can see that the US, Italy, the UK, Spain are all the problematic cases as far as countries are concerned around the world. The beacons have been Hong Kong, not shown here, Taiwan, South Korea, and now we are doing really well as New Zealand. A lesson to us is Singapore and Japan, who originally were well controlled and have loosened the reins a bit, and they have had an upsurge in infections. And as I say, close analysis of this using machine learning will tell us a lot more about how we loosen the controls, the lockdowns around the world, and how we might cope with any recurrence of the epidemic. Okay. In Australia, we have done very well. I think about 26 cases overnight are just reported by Paul Kelly today. And we can see the original control measures prior to the peak of the epidemic in Australia were directed at people coming in from overseas. And then just before the peak, we started to crack down on local spread. And that has been immensely successful. And the problem now is, how do we loosen things? How do we loosen the lockdown? Next slide. The point I want to make tonight in particular is who is most vulnerable to COVID-19. And you can see from this slide, it's clearly the aging. The mortality and the admissions to ICU climb in the aging, and it's remarkable the similarity between the mortality rates in Italy and in China. 15% and 22% in those over 80, 8 and 15% over the age of between 70 and 79, and very little in the youngest people, but most spread occurring between those aged 20 to 40. They're the ones who mix more. Okay. And of course, part of the problem with aging is chronic diseases, comorbidities, cardiovascular disease, diabetes, chronic respiratory disease, and hypertension. We don't really understand whether hypertension is doing this alone or through the diseases it causes. It's definitely not the antihypertensives, at least in China. And immune-compromised patients, Phil O'Connell and Jeremy Chapman have got figures from New York showing that the transplant patients are some of the most susceptible to COVID-19, with mortality rates getting up around 16%. In China and also in the USA, no pre-existing conditions, the mortality rate drops way down. Okay. Now, how does COVID-19 affect the body? Well, it's not just the lungs, and I'll come back to the lungs in a moment. This, particularly in severe cases, is a systemic disease. So the heart is affected, the kidney is affected, people can get diarrhea, and they become, they develop excessive clotting, which leads to stroke and other complications. And that excessive clotting is derived almost certainly from the effects of the virus in the lungs and on the vasculature, the vessels of the lungs. There are two phases to the disease in the lungs: viral growth initially, and then an abnormal immune response. And then the lungs can flood with fluid, and we need a ventilator in the early stages to keep people alive. And in some cases, the lungs become solid, and we may need the equivalent of heart bypass. Okay. So it's clear that early treatment is critical to control the impact of the virus. If you're going to treat the virus, you need to treat early while the virus is still growing and before it induces this abnormal immune response, which is responsible for the severe pneumonia and death. And the most promising antiviral drug is remdesivir, as you probably all heard. It's in trials for COVID-19, both in China on the west coast of the USA, made by Gilead, and the recent US results show shortening of the duration of illness by 31%, but almost, but not quite, a significant effect on survival. And unfortunately, those results have not yet been released because we would like to be able to analyze them. We'd like to be able to see what the exact breakdown of the patients. We do know that these were severe patients that were treated, and we'd like to know exactly how good this drug is if you start early, because that's what most virologists and ID physicians would do, and that is to start the patient early before they get to ICU. Hydroxychloroquine looked promising in a very small open trial, but not in another very carefully designed trial in northern Italy. Its hopes for it are fading, despite some of the 3-page advertisements in our newspapers. Proper WHO and US trials, and one in Australia, are planned, so we'll see very clearly whether it has a role and whether its toxicity is a problem as well. The drugs for HIV have failed in clinical trials published in the New England Journal. Human antibodies from recovered patients are being prepared for trials as hyperimmune globulin, and are also being used as monoclonal antibodies. So we know from the HIV field that you can actually take antibodies from people and prepare them in such a way that they can be much better therapeutic agents than simply just using them as immune serum. Okay. What about vaccines? More than 100 vaccines and candidates are being prepared globally, with 8 of these in clinical trials. Not necessarily the best. The fastest into clinical trials does not mean that they will be the best. As far as we know, almost all of these are aimed at the younger populations, the ones who spread the virus, aged 20 to 50. But if the virus gets through, and Sarah will explain this to you with herd immunity later on, if you don't get enough protection from your vaccine, the virus is going to get through to the aging people in our population, particularly those in aged care facilities. We need a vaccine for the aging as well as a vaccine for the middle-aged. So for the moment, only quarantine and social distancing are available to us, old-fashioned but supplemented by some amazing public health tools. The incubation period of the virus, an average of about 5 days, The upper limit is 12.5, and that's why there's a 14-day quarantine period. Okay. So what's going to happen in the near future? Well, this is going to vary, as I'm sure you all know, by country and by state. Within large countries like the USA, different in Nevada and New York, and it's going to differ by locality. And we can see this when we look at the maps in the Sydney Morning Herald every day. So closely mapping by locality will help contact tracing. Lockdown and social distancing are effective, but there is, of course, a cry for mental health and also the economy to ease as soon as possible. We will still need to maintain social distancing. This is not going to change social distancing, as far as I can see, for at least the next year or more until we get a vaccine. In Australia and New Zealand, we're very fortunate, and we're considering extreme suppression, perhaps even eradication, by extensive testing, especially in hot localities, and detecting all contacts of COVID-19, hopefully electronically, with over 5 million people now downloading the COVID Safe app. Okay. What are we doing at Westmead? We've had more than 290 patients in the Westmead Hospital Isolation Unit or isolated at home. 266 patients have recovered. We made the second and third detections of the live virus in Australia on our precinct in our laboratory, and more than 70 viral sequences from China, Iran, the USA and locally have been done in WIMR and contributed to the international database of more than 600 sequences. Other research initiatives: vaccines for the aging, which I'll talk about in just a minute with Sarah, predictive tests to triage patients who goes into ICU treatment, white cell therapy, looking at the duration of immunity, a really important point. Okay. So looking at what we're doing with vaccine development. Okay. I mentioned before that the aging people over the age of 65 are the most vulnerable. Why is this? Our immunity is declining throughout life, and it really starts to decline still further after the age of 65. Those are the people susceptible to influenza and pneumonia. So the vaccines have been devised more for them than anyone else. Unfortunately, the vaccines for influenza and pneumonia, as I've shown here in the left two bars, are the least effective compared to those in younger people. So only 50%, 55%. Huge breakthrough with the vaccine I've been involved in advising and trialing and chaired one of the publications committee for GSK on, and this is the new Shingrix vaccine, which is more than 90% effective and doesn't diminish in people over the age of 70 or over the age of 80. Quite extraordinary, 90% efficacy over the age of 80. Why is that? It's not just because of the fragments of viral proteins in the vaccine itself. It's because of immunostimulants, or so-called adjuvants. These immunostimulants are there because we no longer have the whole virus, which has bits of fat and bits of nucleic acid in it, which stimulates our immune system. That's what we're used to detecting. So these immunostimulants come out of the cell wall of bacteria and also out of trees, bark of trees. And when one injects this vaccine containing proteins and immunostimulants into muscle, both of them drain into the lymph node, and we know that different immunostimulants will take different pathways through that lymph node, encountering different cell types as they go through, and eventually you get the production of the components of the immune system which are important to control this and other viruses. So that may be antibodies, white cells, some of which kill infected cells, and others of which actually stimulate antibody production. And we need these fragments of virus to go through these different cell types, commander cells, which actually tell the soldier cells which antibodies and white cells to produce. This is critical to reverse the declining immunity of aging people. We need to have the right immunostimulants, but we also need the right proteins and fragments of them. Over to Sarah.
Thank you, Tony. So how do we develop an effective vaccine? What key characteristics should a viral fragment or region of a virus have to make an effective vaccine? The first key characteristic is the viral fragment has to be genetically similar across all viral types. Why is this important? The worldwide COVID-19 pandemic has produced different viral types, and these viral types are genetically varied or genetically dissimilar. So for the vaccine to be effective across the globe, we have to include the viral fragments which are genetically similar. So looking to our figure below, we have established that the gray bar regions are genetically dissimilar across 600 COVID-19 viral types from around the world. And the regions without the gray bars are genetically similar or conserved. So this is a good region of the viral fragment to select, to include into a vaccine that will be effective across the globe and effective for the full pandemic. The second key characteristic is that the viral fragment is essential for viral replication. A very talented PhD student in my group, Yunok Lee, developed a way to determine which regions of viral fragments are essential for viral replication. And this is shown by the blue bars in our graph below. The taller the blue bar is the taller the blue bars are across the viral protein, the more essential the region is for the virus to replicate within infected cells. The third key characteristic is that the viral fragment will be expressed on an affected cell. This will alert the immune system and the cells within the immune system that Tony just mentioned to the fact that this cell is infected by the virus and should be eliminated. Yunok Lee has also developed a way to determine which region will be expressed by the infected cells. And these are shown by the rose bars across the viral protein fragment below. The taller the rose bars, the more the region will be expressed by an infected cell. So these three red highlighted regions below the tall rose bars and tall blue bars, these three highlighted regions of the viral protein fragment would make excellent candidates for a vaccine, as they are genetically conserved across the viral types, essential for viral replication, and will be expressed by infected cells. So by combining these regions with the immunostimulants described by Tony in the previous slide, will most likely help us to produce an effective vaccine for those who are most at risk and most vulnerable for this COVID-19 infection, those who are elderly, and those with comorbidities, excuse me.
Thank you, Sarah. Thank you very much. We're now going to go on to a couple of the questions, and these are some of the questions that we've been asked in advance. So we're going to go to those first, and then we'll go to the audience questions. And the first question we have is on the issue of immunity. Can humans eventually have immunity to COVID-19? And when could we expect a reliable antibody test for COVID-19? So Tony, are you going to take that one?
Yes, thank you very much indeed, Sophie. So antibody tests for immunity in the population, there are over 100 suppliers trying to develop these at present, and some of the biggest suppliers are really starting to push these out. Our own laboratory at ICPMR, led by Jen Koch and Dominic Dwyer, in essence has been doing antibody tests, and we know that they've documented persistence of antibody out to 6 weeks, but we need to know how long this antibody will last and in essence how long immunity will last. It may not just be antibody, it might be white cells or T cells, which we're aiming at. We don't know how long immunity will last and how long resistance to reinfection will last. If you look at the coronaviruses that cause the common cold, they don't make much of an imprint on our immunity. It's only a matter of months. If you look at the first SARS epidemic, in fact, immunity lasted for a couple of years. So we're hoping it's a couple of years at least.
Okay, let's move on to our next question. So I think the next question we have the slide for is, will COVID-19 return? So let's address that. Tony, do you want to take that question?
Sarah will tell you more about herd immunity and how that actually affects the return of the virus. This epidemic has really brought the modelers of infection and epidemics into their own, and the mathematicians amongst us. And so some very good modelers have predicted that we will see probably waves of new infections over the next few years until we get sufficient herd immunity, perhaps in winter because there is enhanced transmission. We heard tonight from Paul Kelly that there's more spread amongst families and also indoors rather than outdoors. And also the virus gets down into the lungs in these droplets because they become smaller in winter in low humidity. So winter is the one we've got to worry about. We need to really keep a close watch on this, I think, next winter. I think we might be all right this winter. And we need to make sure that we don't lapse our social distancing at that time. If we have no antivirals, there's going to be a continuing load on ICUs and ventilators. If we have antivirals, then this will reduce the pressure on the health system that we were hoping would be prevented by a vaccine in the future. And I mentioned before, a predictive test for those who develop severe pneumonia is needed, and that's been worked on by Ben Tang in our institute. And he'll probably tell you more about this in the future.
Sounds very exciting. I'm looking forward to hearing more about that. So let's go on to, you mentioned the issue of herd immunity, but I might just get Sarah to unpack that a little bit more for us. What does the term herd immunity mean? Because I think some people don't really understand what it means.
Thank you, Sophie. Well, the term, there's really 3 models that describe what herd immunity means. Currently, we find ourselves in the first model. And this is a model where no one is immunized, no one has any sort of immunity to the virus, and those who are infected spread the disease quite rapidly throughout the community. That's why social distancing is so key, because by separating people, those who are infected from those who are uninfected, we can prevent a very broad spread of COVID-19. And as we are in this first model where there's no vaccine and no treatment, and the virus is spreading quite rapidly through certain segments of our society and communities, that leads to a lot of mortality and death. And currently, within the world, we have greater than 250,000 deaths related to COVID-19. So we are sitting right now in this first model of herd immunity. In the second model of herd immunity, this is where you may have some vaccines produced, but you don't have a large amount of vaccines available. So these vaccines are then most likely given to those who are at the front line of this disease: doctors, nurses, healthcare workers. So that gives a small group of people within the community some herd immunity, but the rest of the community is still very susceptible to infection. This is also a model for if a vaccine is only about 60% effective. If a vaccine is only about 60% effective, again, some people will be protected, but the majority of the community will be infected by by COVID-19. The third model is where we would like to be. And in the third model, everyone is vaccinated by a vaccine that is about 90 to 100% effective. And that prevents anyone, that allows everyone to develop immunity to the virus and become resistant to infection. And that way, our community is protected from this very difficult pandemic.
Excellent, fantastic. So let's move on to our next question. People are very concerned about how they might contract COVID-19, but there was some early research that COVID-19 could live in the air. Is that correct? And also, how does it live in the air, and should people be worried about exercising, going outside, having someone who might be running sneeze on them? Can you just take us through that briefly?
Sure. So the major mode of transmission is through respiratory droplets. And these respiratory droplets are actually spread by sneezes and coughs. So if a person sneezes or coughs and they're infected and infectious, they can spread the disease to someone nearby by respiratory droplet spread. But we have to remember that within our communities, there are people who are asymptomatic. About 5% of people have no signs or symptoms of the virus. They are infected and they are asymptomatic, and they can spread the virus even though they have no signs or symptoms. Furthermore, before a person starts to, 30 to 40% of people, before they start to show signs or symptoms of COVID infection, they can also shed virus 1 to 2 days before they show these symptoms. So asymptomatic persons and those who are infected with the virus and prior to them having symptoms can also spread the virus through respiratory droplets. Respiratory droplets can be can fly through the air, that is true, but the majority of them drop down to the ground within 6 feet or 1.5 meters. So they drop rapidly to the ground. That's why social distancing is required, that you should stay away from each other or at social distance for 1.5 meters. These droplets can also dry on surfaces, and traces of the virus can persist, but it's unlikely that the virus can be transmitted from these surfaces. Finally, when we think about surgical masks, these surgical masks are very important because they actually muffle these respiratory droplets from being flying through the air when someone sneezes or coughs. So the trajectory of these respiratory droplets can be reduced by wearing surgical masks. So surgical masks are best to be used to block the source of COVID infection, but they don't prevent reception.
And it really depends, doesn't it, Sarah, on the level of community transmission. So for example, in Australia, because the level here is relatively low, that's why the health authorities haven't come out and said everyone needs to wear a mask. Whereas, for example, in like the United States, in states like New York, where the level of community transmission is very high, then the public health message is different. So I think
absolutely.
Tailoring the message based on the audience. So we'll move on to our next question. We've also had a number of questions from the audience about this. So the next question is, how are newborns and mothers affected by this virus? So Sarah, did you want to take us through that one?
Sure. So pregnant women are not at a greater risk of infection than the general population. However, similar to the flu and other infectious diseases, this does cause a respiratory illness and fever, and this can cause serious complications during the first trimester of pregnancy and lead to spontaneous abortions. So although there is no greater risk, there is a risk if a pregnant woman becomes infected and has a fever. So it's best to prevent yourself from being infected. However, high-risk pregnancies, those such as have gestational diabetes, do have a more severe COVID-19 infection. Now, there is no evidence that if a mother is infected, that she can spread the virus to her fetus. However, that's to date. But so far, there is no evidence that the virus can be transmitted through the umbilical cord or the placenta. But we have to still realize that this is a new virus, and there could be additional data coming forward on that. And finally, if a mother becomes infected and she infects her baby, the most common symptom for the infected child is mild breathing problems. So for children, COVID-19 is not that serious of an infection in most cases, but there have been cases of more serious illness in some children.
So, Sarah, while you're talking about children, we have had a number of questions from the audience about children, so I might just get you to address that, including, you know, how safe are schools for children and teachers, and also we've had a couple of questions about asking about the syndrome that's developed in the UK and the USA, where children are developing a syndrome which is sort of like Kawasaki-like syndrome. Are you happy to talk about that? Because we've had quite a few people asking about that
So
on the webinar tonight.
Yeah, that's really true. Kawasaki syndrome has been identified both in England and in the United States, in New York City, which indicates that this virus does not spare any age group. However, this Kawasaki syndrome, it's unclear whether it's caused by the viral infection itself or the inflammation caused by the viral infection. And the majority of the children who have had this Kawasaki syndrome, which is quite serious and can lead to heart defects, have recovered. As far as children shedding the virus, there are cases that children can shed the virus, but I think there still needs to be additional studies conducted to determine how much children do shed the virus.
And the reason, from my understanding, the reason that they feel that it's fine for children to be going to school is the majority of children who've contracted it have contracted it at home from their own families. Isn't that correct? Rather than from at a school environment.
That is true. That is true. But I think, again, this is a new infectious disease, and we may have some more to learn in the future.
Yes, I should just interject and say Christine McCartney on the Westmead campus led the study in schools, and the greatest concern is, of course, the staff in the schools spreading it to each other. Yeah.
And so I guess that's really important to make sure that they keep whatever measures in place to protect themselves. So, Sarah, you mentioned that we're learning more and more about this virus, and that leads us into the next question that I have for Tony, which is, what's the mechanism for the formation of blood clots and the reported occurrence of strokes? Because that's something that we're now learning, isn't it, about this virus, that people are reporting blood vessel changes and strokes, and that's something relatively new. What do you think is going on there? It's
certainly increased clotting. It's large artery strokes in the brain, and we know that there are certain clotting factors that are increased dramatically in people with severe COVID-19. Note that you can also get this in other forms of pneumonia, and about 30% of people with COVID-19 in China, severe COVID-19, actually developed secondary pneumonia. But there's something special about this particular type of pneumonitis. It's associated with this so-called cytokine storm, which is the abnormal immune response, which I talked about earlier. There's also an inflammation of the vessels in the lung, and this gives rise to, particularly to a feature that hematologists know a lot about, and that is a protein called D-dimer. But you'd have to ask my wife about that.
Maybe we'll get her on next time.
Yeah.
So we've got quite a few questions to get through, so I might just get you to keep the answers fairly short, so we can get through as many as we can. But we'll stick with you, Tony. The vaccine project that you've mentioned targeting older Australians sounds very novel. Is there anything like this being done anywhere else in the world that you're aware of?
Well, I've just recently surveyed all of the vaccine initiatives that we can on the internet, and there doesn't seem to be anybody really pushing hard in the aging patients. I think everybody wants to get their vaccines out. It's the surface protein aiming at antibodies. People aren't thinking about white cells and declining immunity. We're the only people in the world who have this model where we can look at immunostimulants in human lymph nodes And also we've got Sarah's remarkable mathematical models that allow us to predict the right fragments of proteins to use.
So just sticking with the issue of vaccines, Tony, how long do you think, what's a reasonable time frame do you think that it could take for Australia to have a vaccine ready for COVID-19?
Well, it's a question of whether it's going to be ours, the University of Queensland CSIRO vaccine, which they're planning for trials in June, or the trials from overseas. Moderna has taken its RNA vaccine into trials. I said there are 8 in trials, including the Oxford vaccine as well. Some of these are good and really look hot stuff. Others don't. And the big vaccine companies are yet to come into this. Once you do, one of the things we've got to be very careful about with vaccines is this aberrant immune response. We don't want our vaccines to induce that aberrant immune response, and this has happened before with SARS-1. So safety is critical. We need to have adequate people recruited into trials to make sure these vaccines are safe. So that means big trials, thousands and thousands of people. And, you know, I don't think we're going to have those trials finished before 2021.
Okay, and I might just ask Sarah, so Tony mentioned about the previous vaccines for SARS-1. Are some research labs looking at the work that has been done with that and maybe a shared protein for SARS-1 and 2 when you're looking for a vaccine for COVID-19?
Yeah, so some labs have looked at what's called the spike protein, and this is the protein on the outside of the virus that actually binds to the cells that it infects. And this protein for SARS-1 has been the protein that's been used to develop vaccines for SARS-1. But this protein is very different between SARS-1 and SARS-2. So the vaccines that have been developed for SARS-1 will not be effective if they're targeting the spike protein. They will not be effective against SARS-2.
So Tony, can you just briefly take us through what are the steps followed to develop a vaccine? Obviously, you mentioned the clinical trials, but that's a very important step. What else needs to happen to get it rolled out to the community?
Well, obviously, there's the exploratory phase where pharmaceutical vaccine companies and academics come up with ideas. And this is what you need. There's 90% of vaccines will fail. It's like a funnel. And bright ideas, there is no monopoly on those. So the more bright ideas around the world, the better we do. Come down through the funnel. Then we go to a series of stages of trial. Phase 1 aims very much at working out whether the trial will induce immunity in a select number of people, small number of people, a little bit about safety, and then we go on to phase 2 trials, which expand things and give us a better idea of safety signals, and then into full-blown phase 3 trials, which look at efficacy. And in the trials I've been engaged in, as many as 30,000 people may be involved in those trials worldwide. The final bit is after marketing, and you make sure that the vaccine doesn't produce a very rare type of side effect. And we've seen a couple of those picked up, very rare over the years, like narcolepsy with an influenza vaccine and reversion of polio to virulence, 1 in 17 million people.
So we might just move on to more broadly just to ask both of you. So what are the 3 most important factors that can support COVID-19 research? Sarah, we'll go to you first.
Thanks, Sophie. Well, in order to continue on with our really technically challenging work, we require funding. So with this new virus and this new pandemic, the funding that we're using to develop our new techniques, our new tools, our new vaccines, all is funding that's not slated for other research. So we really do require funding. And this funding, it would also be used to fund the brilliant minds in our labs that are helping us develop these tools and these vaccines and And so it's really funding. So the three words I would say: funding, funding, funding.
And Tony, what do you think are the three most important factors that scientists really need to support the work of WIMR and to make sure that we can see the developments that you've been talking about actually come to fruition?
Well, I think what Sarah says is true. And of course, we need a coterie of well-trained researchers, PhD students coming through the pipeline. Infectious diseases is that Cinderella of research, in that we, of course, concentrate on cancer and cardiovascular disease and other disease, even kidney disease, Phil. But infectious diseases, we get an epidemic, it comes, we forget about it, another epidemic, HIV, Zika, etc. I think what we need is, just as we've got with networks of diagnostic labs waiting ready to handle this, we need a networked group of researchers throughout Australia. We've got some good institutes ourselves, and Doherty and QIMR, etc. And we need a network of those researchers, better vaccine research capability, and we need continuous funding. And that means government and philanthropy, I think.
Excellent, Tony. Well, look, thank you so much, Tony and Sarah, for all that amazing information. We've run out of time, so I'd like to thank the audience for joining us as well. But before you go, if you would like to support the great work that both Tony and Sarah and everyone at WIMR are doing, there are two things that you can do. You'll all receive a summary of tonight's presentation, and you can get the word out about the work that WIMR is doing and spread it through your social media channels. Tell your friends and colleagues about the great work that's being done. Secondly, if you're able to, WIMR would love you to donate directly to their work, and you can do that by going onto their website. You can see it's on the screen now. It's westmeadinstitute.org.au, and you can also find out about gifts, corporate partnerships and bequests as well. So thank you so much for being here. It's a bit different in this virtual world, but there's no doubt that we've been able to get across some amazing information this evening from some spectacular minds who are doing such important work. And WIMR plans to do more of these Meet the Researcher evenings. And so we hope that you'll join us at any upcoming events that we do have. And hopefully down the track, when the physical distancing is loosened, we might be able to meet again in person. But until then, stay in touch, and we look forward to seeing you at our next event. Thanks for your time this evening. Good night.
Thank you, Sophie. Thank you, Tony. Good night.
Thank you, Sophie and Sarah.